Hello students. Today we are going to discuss Unit 11 Shrimp Hatchery Technology from BSc third year, semester 6. So the objectives of this chapter are we will be learning the importance of shrimp hatchery technology and their life cycle. We will also understand the preparation of brood stock, selection of spawners and egg collection. We also understand hatching and transportation of nopli and their rearing. We know the hatchery management and we also understand harvest and transport of larvae. So to introduce this topic, so traditionally shrimp fry are trapped and held in ponds and later collected by shrimp gatherers for stocking in grow out ponds. With increasing demand for shrimp, supply of wild fry for the increasing number of shrimp farms has become insufficient and inconsistent. The breakthrough in the completion of the life cycle of commercially important shrimps in captivity, such as the tiger shrimp, the Japanese kuruma, the eastern shrimp, and the banana shrimp, has greatly enhanced mass production of shrimp fry under hatchery conditions. The excellent growth performance of these hatchery bread fry in grow-out ponds strongly shows that the shrimp hatchery can answer the industry needs for ample supply of shrimp fry for farming. From many years of accumulated experience and research findings, the success of a shrimp hatchery depends on the choice of a suitable site, effectiveness and efficiency of the hatchery design, experience of hatchery technicians, efficiency of operational management. Then, how to select a suitable site for the hatchery? We need to see various conditions, water supply, everything. So, the seawater used in hatchery should be clean, clear, and relatively free from silt. The water quality should be stable with minimal fluctuation in salinity. Suitable sites are usually found in sandy and rocky shore ecosystem where there is clean, clear and good quality seawater all year round. Sites not suitable for hatchery or swamps and muddy shores where the water becomes turbid during heavy rains or due to turbulence. We need to avoid the river mouths where abrupt lowering of salinity often occurs during heavy rainfall. And what is the advantage of rocky and sandy shores is that good quality seawater is relatively near the shoreline, thus reducing the cost of piping installation and pumping cost. And the hatchery site should also be free from possible impact from any inland water discharges containing agricultural or industrial waste. Then we need to see the availability of spawners also. Presence of spawners at the vicinity of the proposed site is of considerable advantage in ensuring consistent supply of spawners, reducing the cost of transportation, which could affect the rate of spawning. Then we need to consider the availability of power source also. Electricity is essential to provide the necessary power to run equipment and other life support systems in the hatchery. Though some marine pumps and aerators can be driven directly by handy generators, the shrimp hatchery can therefore be operated in areas without electricity supply. However, it is more economical to operate it in areas where there is a reliable source of electricity. So, installation of an on-site standby generator is absolutely necessary, especially in areas where there are frequent lengthy failures and fluctuations. So, when there is power cut, we should be readily prepared by using the generators and all. Next, we need to see freshwater supply. Freshwater is essential for daily hatchery operation such as salinity adjustment, equipment maintenance, and for domestic use. Then next one is accessibility. Ideally, a hatchery should be sited in areas where there are active shrimp farming operations so that the shrimp larvae produced can be easily transported and distributed to the grow out ponds. So, the site chosen for hatchery establishment must be easily accessible to facilitate communication and transportation. Then we need to look into the climatic conditions also. A hatchery can be established in any climatical conditions as long as the required rearing conditions can be adequately provided. However, all the commercial hatcheries take full advantage of nature in terms of energy source and supply of good quality water. Sunlight is the basic requirement for hatchery operations, especially in the mass production of natural food used as feeds for the growing shrimp larvae. In the temperate region where adequate sunlight is only available in certain periods of the year, hatchery operations are usually confined to a certain suitable season. This is especially in the case of some marine shrimp such as Penis japonicus and Penis orientalis, which have a single pronounced spawning season. In such situations, hatchery production is relatively limited to a short period of time. On the other hand, the tropics are endowed with warmer climate and adequate sunlight all year and have ideal climatic conditions for hatchery operation, especially for many warm water species such as the tiger shrimp, Penis monodon, the white shrimp, Penis indicus, which spawn all the year. And the optimal temperature for hatchery design may be necessary in areas where there are long period of rainfalls, which often reduces the intensity of light, cause turbidity in water supply, and temperature lowering of water temperature. We need to see the hatchery design and how it has to be constructed. Production target can be determined based on a market demand and financial input. In the case of species such as Penis monodon, at which the production of fry depends on the availability of spawners from the wild, production target is limited by the supply of spawners. This limits the production capacity of the hatchery, whereas Penis japonicus and white shrimps at which spawners are easily available, production capacity is unlimited. In that case, the tank capacity up to 2500 cubic meters can be seen in many large scale hatcheries in Japan, where Penis japonicus is the primary species grown there. However, in most Southeast Asian countries where tiger shrimp forms the target species for hatchery production, so the tank capacity is considerably reduced due to limited spawner supply from the wild. Hatchery design is aimed at achieving the production target which determines the size of the hatchery. And the tank capacity is based on an approximate ratio between algal culture tank and larvae rearing and nursery tanks. Desirable algal tank capacity is 10 to 20% of the larva rearing tank capacity. The capacity of maturation tank depends on the number of spawners needed. Then, what is the size of the hatchery? Generally, the size of a hatchery should be based on its functional requirements and economic efficiency. Based on the level of operation, production output, and financial investment, hatchery practice can be broadly grouped into three categories. One is small scale, medium scale, and large scale hatchery. Small scale hatchery. This is a backyard hatchery, usually owned and managed by the shrimp grower himself, using family members or immediate relatives for additional labor. The main goal is to supply his own shrimp farm with the required number of fish fry, and the excess may be sold to the neighboring growers. Usually, the hatchery site is an extension of the farmhouse with floor space ranging from a few square meters up to about 1,000 square meters. In such hatchery operation, the total production capability seldom exceed 5 million post larvae per annum, and the hatchery is operated by not more than two technical personnel. Medium scale hatchery. This type of hatchery is relatively larger than the small scale hatchery in terms of capital investment, hatchery size, production capability, and scale of operation. While hatchery management is somewhat similar to that of small scale hatchery, the production capability range between 10 to 20 million post larvae per annum and is operated by about 3 technical personnel and 3 to 4 laborers. This type of hatchery is usually put up by small cooperatives to supply the required shrimp fry to its member growers. Private entrepreneurs or government agencies may also establish hatchery of such operational scale to produce fry of sale or distribution to the growers. Then if you see the large scale hatchery, this scale of hatchery is commercially run by big corporations, national agencies or cooperative projects. While the capital and operational investment far exceed that of the medium scale hatchery, production capability of such hatchery usually exceeds 20 million post larvae per annum. Such hatchery is centrally and symmetrically managed and is supported by a pool of not less than 6 technical personnel and 6 to 10 laborers. Then what should be the facilities at the hatchery? In designing a hatchery, ample space should be provided for the rearing and support facilities needed in the operation. A functional hatchery should have the following essential components. They are first one, maturation tanks. What are maturation tanks? The major constraint in hatchery operation of tiger shrimp is the limited supply of spawners from the wild. Hence, eye stalk ablation techniques can be used to augment the scarcity of spawner supply. Thus, maintaining ablated shrimp in maturation tanks would ensure a constant supply of gravid females. The shape of the maturation tanks can either be circular, rectangular, or oval. The tank capacity may vary from 5 to 40 tons with depth ranging from 1.2 to 2 meters. If the shrimps are kept for less than 5 weeks, bottom substrate is not needed in the tank. The tank is installed with an inlet pipe from the wall and a double cylinder standpipe at the center for drainage. This system facilitates continuous flow through of seawater. Next, the spawning tanks. Spawning tank should be circular with a flat or conical shaped bottom. Water holding capacity may vary from 50 liters to 1.5 tons. The tanks can be made of fiberglass, plexiglass, plastic, or marine plywood. The tanks are used to temporarily hold the gravid females until spawning. This is the picture where it shows the cross section of 12 tons ferrocement broodstock tank. Next, coming to the larval rearing tanks. Two types of rearing tanks are being used to rear the newly hatched larvae. In Japan and Taiwan, larger tanks with a capacity of more than 50 tons are being used. In Southeast Asia, most of the hatcheries use smaller larval rearing tanks of about 3 tons capacity. Hatchery operators call the later system of larval rearing as the small tank hatchery system, which originated from the Galveston Laboratory in the USA, and the farmer system as the large tank hatchery system, which originated in Japan. Small tank system. The larval rearing tank may be circular, rectangular, or oval in shape with tank capacity ranging from zero point 0.8 to 3 tons. The bottom circular tanks may either be flat or conical. Rectangular or oval shaped tanks always have flat bottom. The circular tank is usually 1.8 meter in diameter and 1.2 meter in depth with a central double cylinder standpipe drainage system which can be used for continuous flow of seawater when the larvae reach mysis or post-larval stage. Rectangular tank is about 1.5 into 5 into 1 meter in size. The drainage pipe is set at the side of the tank. The drain pipe is also used for harvesting. In all types of tanks, seawater is delivered into the tank through an inlet pipe installed at the top of the tank. Then big tank system. The tanks used are rectangular or square in shape with capacity varying from 50 to 2000 tons or more. The tanks can either be located outdoors or if located indoors, the transparent roofing should be provided to allow for sources of sunlight. In a big tank system, spawning, hatching, and larval rearing operations are done in the same tank. The larvae are reared for 30 to 40 days. Then water storage and filtration tank. The water storage tank is normally elevated to effectively distribute water by gravity to the hatchery. The water storage tank capacity should be at least 20% of the larval rearing tanks. Storage tanks are normally constructed out of reinforced concrete to withstand the water pressure. When the water is turbid, installation of a filtering screen and sand filter unit becomes necessary. The filter chamber may be constructed adjoining the holding tank. There are two types of filter systems. Gravity filter, where water is pumped into the filter chamber over the surface of the filter bed and allowed to pass through the filter material by gravity to the holding chamber, which is located under the filter chamber. And reversing filter, where water is pumped directly to the space under the filter chamber and pumped upward through the filter to the surface and onto the holding tank. In both systems, the filter chamber usually contains white sand, charcoal, gravel, or all the three as filter material. The advantage of the reverse system is that water passes slowly through the filter material and the whole surface area of the filter is utilized. It is easy to backwash by spraying water from the surface of the filter and the detritus underneath are easily washed out. On the other hand, gravity filter method allows water to pass through the water chamber too fast and do not fully utilize the surface area of the filter unless it is provided with pipe spray water over the whole surface area. The disadvantage of the gravity filter system is that the filter is easily blocked with detritus after a few days of operation, resulting in turbid water and difficulty in backwashing. Then aeration. Aeration is essential during the entire larval rearing process in maintaining sufficient dissolved oxygen concentration in the water, ensuring even water temperature throughout the water column through turbulence and also help reduce the ammonia content in the water. Large volume of low pressure air, while an air compressor provides small volume of high pressure air. An air blower runs continuously while a compressor, which is equipped with the pressure tank, runs whenever the pressure is low. The compressor automatically switches on when the pressure drops below a predetermined level. In a hatchery, low pressure air available at large volume is more desirable than high pressure air at small volume. Moreover, the hatchery tanks are seldom more than 2 meters deep. Rotary air blowers are not designed for oil free operation and have a tendency to blow oil particles into the airline, producing oil slicks on the surface of the water. Air filters at the inlet and outlet pipes are therefore needed. When the capacity of an air blower is less than 10 HP, ordinary air filters for automobile may be used at the inlet pipe. However, for blowers with high horsepower rating, synthetic foam can be used. Adjustable pressure tank with resin glass beads or bagasse as filter materials are used for the outlet pipes. The Roots blower is more appropriate for hatchery purposes because it seldom breaks down, less complicated to use, and does not produce oil slicks. In culture tanks with depth less than 2 meters, an air pressure of about 0.2 to 0.3 kg per cubic centimeters and a volume of 4 to 5 liters per meter square per minute is sufficient to oxidize the dissolved organic matter in the tanks. Since continuous aeration is essential to the survival of larvae in high density, any prolonged power interruption would seriously affect the culture organisms in the tank. Thus, it is essential to install an automatic switch which starts a standby generator whenever there is a power failure. A battery operated warning device to signal the crisis and the required operation of the standby generator can also be used. Seawater supply and piping system seawater can be drawn directly from the sea or from a sump pit. If the course of the water is relatively clear, the water can be pumped directly into the overhead filter tank and stored in the reservoir or storage tank. Water is then gravity fed to various culture tanks through its delivery pipes. However, if the water is turbid and contains a high concentration of suspended solid, the water is pumped first into a sedimentation tank where the suspended solids are allowed to settle down and the clearer water on the top is pumped into the filter tank. Sometimes, when the water source is far from the shore due to low tide and if large quantity of water is needed continuously, the intake pipe should be laid horizontally from the littoral zone to the underground sump pit and the filter tank. Whenever possible, the seawater should be drawn directly from a tube well. Water from the tube well is usually clear and clean water is needed. It can be pumped directly into the filter tank. Then marine pumps. Centrifugal and submersible pumps are commonly used in hatchery operation. Centrifugal pump is more desirable in the big hatchery because it has a higher total head capacity. The selection of the size and type of marine pump depends on the size of the hatchery and the daily water requirement. For the smaller backyard hatchery, a submersible pump with a discharge pipe diameter between 1 inch to 4 inch and a discharge capacity of 4 to 20 tons per liter is sufficient to provide the water needed. Medium and large hatcheries normally use centrifugal pumps. The size of the pump depends on the total water requirement per day, and the maximum pumping time. Then the construction and the layout. Once the project site is selected and the production target defined as an aerial survey of the proposed site will help to determine the perimeter of the area. An aerial view would show the important topographic characters such as rivers, shoreline, mountain, and low-lying plains. A master plan of the hatchery is then made. Layout of the hatchery should provide a schematic design of the location and integration of various facilities such as buildings, broodstock tanks, larval rearing tanks, nursery tanks, spawning tanks, pump house, air supply and powerhouse, laboratory, staff house, piping for water supply, and drainage canal. The layout plan should include the exact dimensions, locality, shape, and size of the said facility. Technical drawing should show the detailed structure of the facility and size and quantity of support materials needed to ensure stability of the facility. From the technical drawing, the owner can roughly estimate the cost of construction. Since the technical drawing presents details of the structure such as thickness of the tank wall, amount and size of iron bars, and maximum proportion of concrete, the price of these materials will be easy to canvas to arrive at the cost estimates. However, estimations of construction costs should be done item by item before making the total estimation. In the total estimates, labor cost should be also included. If you see the life cycle of the spawn, based on their mode of life, pinnate shrimps can be grouped into wandering and burrowing groups. Wandering group means species belonging to this category have pronounced age groups from dense schools and are always on the move and active day and night. They prefer turbid waters with soft mud bottom, burrowing slightly or not at all into the substrates. A typical wandering species, Penis orientalis in the Yellow Sea, migrates southward for overwintering and northward in spring to its spawning ground. The distance between the areas for overwintering and spawning is approximately 700 to 1300 kilometers. Other species belonging to this group are Penis setiferus of the Atlantic coast, Penis morganensis, and Penis indicus of the Southeast Asian region. These tropical species, however, do not need overwintering. Penis morganensis and Penis indicus generally spawn at a depth of 7 to 30 meters in offshore waters, usually near the nursery ground. The larvae spend part of their life cycle in nursery ground until adolescent stage when they start migrating to deeper and more saline waters. Since Penis margensis, Penis indicus, and other wandering type of pinnate shrimps from dense schools, fishermen can easily collect large number of spawners during peak seasons. Next, coming to burrowing groups. This burrowing prefers certain habitats with sandy bottom. They do not have pronounced age groups. They show marked nocturnal activity, burrowing into the bottom substratum during the day and emerging at night to search for food. This diurnal activity is closely associated with changes in light intensity but may also be due to other factors. Examples of this group are Penis japonicus in the Japanese coast, Penis durarum of Florida, and Penis monodon in Southeast Asia. Penis monodon spawners are found in offshore waters on sandy bottom at depths of 20 to 40 meters. The larvae move towards the coast, enter estuaries and mangrove swamps which serve as nursery ground. They then migrate to deeper water when they become adolescent and finally move to spawning grounds upon becoming adults. Peanut shrimps generally spawn in offshore waters. Each gravid female layers as many as half to approximately three quarter of a million eggs per spawning. After hatching, the early larval stages like Nauplius, Protozoa, Mysis, and early post larvae remain planktonic for some time and are carried towards the shore by tidal currents. The first larval stage is known as Nauplius. The unsegmented body, which is pyriform in shape, possesses three pairs of appendages. The Nauplius undergoes six moltings within 50 hours into a protozoa. The body becomes elongated with a distinct cephalothorax. The early protozoa stage has a pair of protruded compound eyes. The next stage is characterized by the presence of a rostrum, and the late protozoa stage has a pair of uropods. After 4 to 6 days, the protozoa finally metamorphoses into a mysis. At this stage, the larva assumes the form of a juvenile shrimp, or which the pleopods are to develop. At this stage, tiny protrusions known as pleobases are seen on the ventral side of the abdominal segment. The next stage is marked by the development of first segment of the pleopods development. These myces remain drifting in the water column until they metamorphose into post larvae within 10 to 12 days. At post larval stages, the pleopods become fully developed and functional. The animals grow very fast in terms of size and are able to swim freely, although early post larvae are still planktonic in offshore waters. At a body size of 0.8 to 1 centimeter in body length, they enter into the estuaries and inshore shore waters, where they first adopt a benthic existence. The shrimps spend their juvenile, adolescent, and sub-adult stages in estuaries where estuarine waters, and then gradually move towards deeper water as they grow and eventually returning to offshore water when they obtain sexual maturity. Inadequate supply of spawners remains as one of the major constraints in the development of the shrimp farming industry, especially for species such as the tiger shrimp, Penis monodon. Recent success in eye stalk ablation techniques has greatly enhanced constant supply of spawners through oblating captive brood stock to maturation. Conditioning of brood stock. Upon arrival in the hatchery, the brood stock are first acclimatized in holding tanks for 4 to 7 days. The holding tank should be big enough to provide proper space and aeration. 60% of the water tanks in the tanks is changed daily. Once the animals have recovered from transport stress, they are induced to molt by manipulating salinity of water in the holding tank. The salinity is decreased by about 4 to 5 ppt for 2 days and then increased to the normal salinity of the seawater. Majority of shrimps will molt after changing salinity. Mating occurs during this time when the females are newly molted. The shrimps are then ablated to 2 to 3 days after molting or when the shell is completely hardened. What is induced maturation? Only suitable broodstocks are used for eye stalk ablation. The criteria are first one complete appendages, second one presence of spermatophore in the live in the thylicum of females, then size should be least by 100 grams. Induced maturation. Only suitable broodstocks are used for eye stalk ablation. The criteria are complete appendages, presence of spermatophore in the thylicum of females, size should at least be less than or least 100 grams. Presently, the most practical way of induced maturation is by unilateral ablation of either right or left eye stalk of the female. Ablation is done by using a razor blade to cut or open the eye, then squeezing out the eye stalk from the base to the tip when the thumb and the forefinger are using the fingers alone to break and squeeze the eye. The ablation animals are stocked in maturation tanks at a density of 5 to 6 meters per square meter and a sex ratio of one male to one female. Then maintenance of broodstock in maturation tanks. The broodstock are fed with squid, mussel, or cockle meat or pellet feeds at the rate of 10% of total biomass. The water in the tank is allowed to flow through continuously or changed daily at 6 to 70 degrees of total capacity. Sampling. Gonadal development of an ablated female is checked 3 to 5 days after ablation while checking for gravid females is carried out every other day. Sampling and checking are done at tight or at any time if the tank are sufficiently covered and kept dark. During sampling on under underwater flashlight tied to a pole is held close to the shrimp so that the light strikes perpendicularly on the dorsal part of the body where the ovaries are located. Water in the maturation tank can be lowered to 30 centimeters to allow the workers to get inside the closer observation. Only gravid females with stages 3 and 4 ovaries are collected and transferred to spawning tanks. Larval feed. Under natural conditions, peanut shrimp are either omnivorous scavengers or detritus feeders. In general, shrimp larvae feed on phytoplankton, detritus, polychaetae, and small crustaceans, and their food preference changes with age. They start feeding of protozoa stage. Protozoa and early mysis stages prefer phytoplankton. At mysis and early post-larvae, food preference changes to zooplankton such as rotifer or brine shrimp. As the larvae grow older than P6, feeding habit changes again to that of a bottom feeder. Polychaetes, chopped mussels, or cockle meat are fed during these stages. Large scale production of phytoplankton for larval rearing can be obtained in two ways: by direct fertilization of seawater in the rearing tanks or from pure culture. Many workers in the field rely partly on mixed populations of phytoplankton present in rearing water. In the past, fertilization of tank water was done directly with the resultant algal production from the blooms utilized for feeding purposes. The method, however, has its disadvantages. Using direct fertilization techniques, heavy blooms of undesirable species often occur. Such circumstances have led to the development of techniques for screening different algal species suitable for larval culture and legislation and isolation techniques for pure culture of specific types of phytoplankton. In addition to these, many other types of feed have been developed and tested as part of peanut shrimp larval rearing strategies. These feed may either be frozen or dry material from the mollusks. Crustacean tissue, soybean cake, soybean curd, egg mustard, and fertilized eggs or oysters. Other types are available as formulated diet or the newly developed microparticulate and encapsulated diets. However, the choice of a particular feed used should be properly evaluated based on the following criteria. One is availability and ease of handling, then feed performance, then feed cost, and then rate of return on capital. Then phytoplankton culture. The more popular phytoplankton species found to be suitable as food for the early larval stages of shrimp are Chetoceras calcitrans, Skeletonema costatum, Tetraselmis species. These plant components of the oceans usually require certain environmental conditions for growth. In the wild, phytoplankton abundance is easily affected by fluctuations in temperatures, day length, presence of grazers, availability of nutrients, water depth, and turbidity, as well as the seasons of the year. Manipulations, however, of the above mentioned factors is easily possible under laboratory conditions or control rooms. Phytoplankton culture is usually carried out by subjecting a known amount of phytoplankton to an environment suitable for its growth. How to cultivate rotifers? The rotifer Brachiones plicatilis is the most important zooplankton utilized as live food for various cultivable marine animals. Culture of rotifers usually involves the use of various algae like chlorella and tetraselmis, as well as other food items like bakers and marine yeast. Brine shrimps as larval feed. Many hatcheries depend largely on brine shrimps to feed their growing shrimp and fish larvae. In most cases, it is the knockley that are used to feed the larvae, probably because of its small size and relatively slow moving habits. They can be easily preyed upon by the predatory fish or shrimp larvae. Preparation of egg custard contrary to the use of boiled egg yolk. So preparation and use of egg custard is preferred for use as larval feed. Chicken egg is broken open and the egg white and yolk placed in a bowl. Add a small volume of water while manually stirring the solution, or mixing can be done with the use of an electric blender. Then steam the homogenized solution until it hardens. The egg custard is then passed through a sieve with a desired mesh size. Weigh the particulate egg custard according to the desired quantity, that is mg per number of larvae. Contrary to the use of boiled egg yolk, preparation and use of egg custard is preferred for use as larval feed. Chick- then selection of spawners and egg collection. Although spawning occurs throughout the year among the tropical species of shrimps, there are distinct periods of the year when majority of the shrimps spawn. In the case of Peneus monodon in Southeast Asian waters, there are two pronounced spawning seasons from December to March and June to September, with only one pronounced period from June to September in the case of Peneus margensis and Peneus indicus. While spawners of these species of shrimps are supposedly available all the year, they are abundantly caught during the spawning season. The gravid female can be easily recognized by the presence of a very large and distinct diamond-shaped dark green mass of ovary between the first and second abdominal segment below the dorsal shell. Selection of spawners can be easily done by holding the animal with its ventral body facing a light source. The criteria used for selecting spawners from the wild are stage 4 ovary, complete appendages, the back is not broken, presence of spermatophore underneath the thylicum, the color of the shrimp, especially Penis monodon, should be pink with a faint greenish tint. Spawners which are slightly reddish could be due to stress caused by abrupt lowering of the water temperature during transportation by fishermen who try to delay spawning. Stressed spawners give very low spawning rates. Then procurement and transportation of spawners. The major fishing gears in collecting spawners can be broadly grouped into three categories: stationary trap, mobile, and entangling net. Then treatment of spawners, spawning capacity, egg collection, and treatment. After spawning, the animal is removed from the tank the following morning. The eggs are then cleaned either by siphoning into egg collectors or draining 2 by 3 of the water through a filter net that effectively retains the eggs within the tank. When draining is completed, the scum is then removed using a scoop net with a mesh size bigger than the shrimp eggs. The tank is then filled up with new seawater. During the cold season, fungus and bacteria are likely to infect the eggs during incubation. Preventive treatment normally consists of dipping the eggs in 1 ppm of methylene blue or 0.5 ppm of malachite green for 10 minutes, or else 3 ppm KMNO4 for 30 minutes. After that, the eggs are transferred to a cleaner tank for further incubation and subsequent hatching. From the incubation hatching tank, samples of eggs are counted to determine the number of eggs spawned per female. Then hatching and transportation of napli. Eggs of most species of shrimps within 12 to 18 hours after fertilization at temperature and salinity range of 26 to 30 degrees centigrade and 30 to 23 ppt respectively. The density of napli is estimated a day after hatching. Napli from 300 ml water samples taken from the spawning tank are counted and averaged. So the total number of napla in the tank is then obtained by the multiplying the volume by the average density. Then coming to the transportation of napla. Plastic containers. By using the plastic containers, only strong and healthy larvae should be transported. This is done by concentrating the napli at the water surface with a light source at night. Gather them by scooping with a plastic or glass container. The larvae are then transferred into plastic jars which are half filled with seawater. The container is then gradually filled up. A 20-liter plastic container can contain a maximum number of 5 lakh napli. The open end of the container must be properly sealed to prevent leakage. The survival rate after 6 to 8 hours during transportation is more than 50%. Then by using plastic bags. Each bag containing about 6 to 8 liters of water can be stocked with 2 lakh napli. The water in the bags is oxygenated and the open end is closed with rubber bands. The survival rate is about 80 to 90% if transportation takes about 4 to 6 hours. After the transportation, routine hatchery management. So the maintenance of optimal environmental conditions is necessary for maximal growth and survival of the cultured organisms. So we have discussed the importance of maintenance of water quality. What should be the quality of the fresh water and how the seawater should be supplied? How water quality and the quantity should be maintained. Then we also understood how to maintain the salinity of the water. If it is seawater, what should be the salinity? If it is the fresh water, what should be the salinity? And the temperature also, what should be the minimum and maximum temperature and how to control the temperature and what time the temperature should be controlled also. Then we need to take care of the dissolved oxygen. What should be the percentage of the dissolved oxygen? We should also know if the dissolved oxygen is less, what needs to be done. If it is more, what needs to be done. Then we should have a check on pH and nitrogenous compounds that are present in the water also. The pH should be, it should neither be too high or not too low also. It should be maintained at the correct level. So with this, we have come to the end of the chapter. Let us see what type of questions are asked from this chapter. These are the model questions. Answer the following in about 30 lines each. Write about selection of a suitable site for a shrimp hatchery. Write about shrimp hatchery design and construction. Write about hatchery facilities. Write about seawater supply and piping system. Write about life cycle of wandering group. Write about life cycle of burrowing groups. Write about preparation of brood stock for spawning. Write about shrimp larval feed. Write about procurement and transportation of spawners. Write about hatching and transportation of nopli. Write about shrimp larval rearing. Write about shrimp larval rearing in small indoor tanks. Write about shrimp larval rearing in large nursing tanks. Write about maintenance of water quality in shrimp hatchery. Write about shrimp nursery of post larvae. Write about harvest and transport of shrimp larvae. Thank you.